an exciting talk. Welcome, David. Thank you. We're listening to your talk.
Very good. Thank you. So the path to protocells must involve synthesis of polymers, and that's part what this discussion has all been about. We're interested in non-enzymatic synthesis of nucleic acids, and it's been going on now in our lab for perhaps 15 years. I've had some active collaborations with Bruce Damer, Tuwe Hassankam, and David Ross, and you're going to hear some of those results in my talk. I've had previous collaborations that contributed to this, and in fact, Sarah Maurer, our keynote speaker, was among those that contributed in such a collaboration. We are currently interacting with the astrobiology group in Australia and New Zealand. That's Martin van Kranendonk, his grad student, Luke Steller, and Catherine Campbell in Auckland. So we have guiding questions, and they're very straightforward. Where did nucleic acids come from? And you might think that's simple, but we're talking about 4 billion years ago. So this is a very different question. Nobody has an answer yet. We are stepping toward answers. That's what you're going to hear about today. This must have involved some non-enzymatic polymerization that went on in the prebiotic environment. And the question is, how did the polymers begin to replicate? And that's what Zebran just talked about. He is interested in the replication process. This is sort of the edge of our knowledge. I can take you up to that edge, and then we just have to leave that for future work. So let's first consider polymers. The ones that we're most interested in are the two kinds of polymers, proteins and nucleic acids. There were no enzymes or metabolic reactions in the prebiotic environment, and so we need to think about conditions that had a relatively simple process. We still don't know quite what that is, but it's got to be simpler than what is happening today. And that's what we're looking for. And the question is, where could it happen? So our thought is that one possibility is freshwater hydrothermal fields on volcanic islands emerging from a global ocean on the early Earth. These have the property of evaporating and refilling indefinite numbers of time, and that wet-dry cycle can add chemical energy to any potential reactants and help polymerization along. Well, I wasn't ready to just stay in the laboratory and think about this. I wanted to see what this looked like. So we've been visiting what we call prebiotic analog sites. This began way back in 2004 with a trip to Kamchatka. This is far eastern Russia. It's a peninsula just north of Japan. And I did a simple-minded experiment and said, here we have a boiling hot spring. Let's see what really happens if we put a prebiotic soup composed of amino acids, nucleobases, one amphiphilic molecule. We chose myristic acid, which is a 14-carbon fatty acid, along with glycerol and phosphate. The pH is mildly acidic, pH 3, that's produced by the acid gases that are part of most volcanic emissions. What we found was quite interesting. We discovered, and there's a real surprise, that most, but not all, of the components of that mixture adsorbed to clay particles circulating in the pool by this boiling action you see in the center. The one exception was the fatty acid. As you might expect from a soap-like molecule, this produced a froth around the edges of the pool, and that froth, in fact, is composed of self-assembled bilayer membranes. And that really was the start of the work that we're doing now. What would happen to a mixture, a prebiotic mixture, as it went through wet-dry cycles in small freshwater pools? at low pH. So I took that idea back to Santa Cruz. Oh, by the way, this is published if you're interested in the philosophical transactions, 2006. So I went back to Santa Cruz and I wanted to simulate what I thought were favorable properties of hydrothermal field environments. Organic compounds would obviously accumulate in hydrothermal ponds rather than diffuse into a vast seawater of an ocean. They would be in an acidic pH range, but not too acidic. The concentration of ions, and we looked at half a dozen hydrothermal pools around the world, is down in the millimolar range. This really is close to being distilled water, and in fact it is water distilled from the nearby sea and falling as fresh water to feed these pools. These pools are going to go wet-dry cycles that can concentrate potential reactants. Polymerization, I'm going to show you, is promoted in the dry phase of a cycle. Elevated temperature provides activation energy. These are up in the range of 80 to 90 degrees C. And finally, if a lipid is present that can form membranes, the lipid encapsulated any products, polymeric products of the reactions to form what we call protocells. So this is different than the hydrothermal vents that you heard about yesterday. I treat this as an alternative hypothesis. The hydrothermal vents and the hydrothermal fields are open to experimental testing, and I'm going to show you some of the tests that we've been doing in the laboratory, but also out in the field. We have some experiments going on in actual hydrothermal conditions. So this is a simulation chamber we built in Santa Cruz. It's got a rotating disc that is maintained at around 80 degrees C. It rotates very slowly, about 7 degrees every 15 minutes. And this is driven by a stepper motor that you can't see. It's underneath the disc. Heat sources, just ordinary laboratory hot plates. Carbon dioxide fuses or diffuses, actually is under pressure going into this chamber. And that's a drying portion of the cycle. So it goes through 4 little ports on either side of that disc that you can see. There's also a hydration cycle, and a typical reaction mixture is about 1/10 of a mil that goes through basically as many cycles as you want. Typical cycle might be anywhere from half an hour per hydration cycle up to about 2 hours. So something that you've got to understand is that these conditions can synthesize ester bonds. So any organic chemist knows this reaction. It's one that we learn in organic chemistry, and probably this was my first organic chemistry experiment that I did as an undergraduate at Duke University. If you mix ethanol and acetic acid, there is a spontaneous reaction driven by the chemical potential available to form an ester bond, and water comes off as a product of this. It goes to equilibrium, except that you can do one more thing to pull it away from equilibrium. I'm going to show you that in just a moment. What you want to keep in mind is that this should also work with mononucleotides. Adenosine monophosphate has a acidic phosphate group. The neighboring molecule has a hydroxyl group, and basically an ethanol, like ethanol. This can form a ester bond if we can pull the water away. And that is what happens in both of these conditions. To make the ethyl acetate, if you want to make more of it, you pull the water out of the reaction so that the reaction goes to the right. And what we're doing in our condition is evaporating water, and it turns out that that provides chemical potential that'll pull the reaction to the right with extensive synthesis of long strands that are, in fact, nucleic acids. So let's take a look now at some of the kinds of experiments we're doing. We have the ability to sequence molecules by nanopore sequencing. The simplest molecule to work with are deoxymononucleotides. So I'm going to show you one unpublished experiment we've done in which we have synthesized DNA by wet-dry cycling. We start with thymidine monophosphate. And by the way, Christian Meyer yesterday said he thought that this takes an activated molecule. It does if you're doing this in solution, but that's not required by a drying cycle that provides the chemical potential required for polymerization. So we put the TMP through multiple wet-dry cycles. We precipitate it as oligothymidyllic acid. We then ligate that to a known DNA, and the reason for that is that these are strands that are too short for the nanopore sequencing, so we have to ligate it to a longer strand. Then we precipitate it again and do nanopore sequencing. I want to show you what nanopore sequencing does. These are devices now available through Oxford Nanopore technology. The one that we use is a MinION. You can buy it for about $1,000 and do what we've done here. There is a helicase that unzips a double strand of DNA at about 450 nucleotides per second. The single strand is pulled through a pore by a voltage. It ratchets through, and you can see then changes in the ionic current that are base specific. Those are picoamp modulations, and the instrument now is able to translate that into a sequence of bases. This is what a MinION looks like. We put in about 200 microliters of our processed DNA, feeds into a laptop, and if you're watching the screen, you can watch individual molecules of DNA going through the nanopores in this device. Here is a gel of the TMP oligomer. Clear to the left, you'll see radioactively labeled ladder, all the way from 30 mers to near 100 mers. We've scanned it, and just to the right of that, the white on black trace shows the scan of that gel. We then look at the base sequences, and as expected, we do see oligo T as part of the base sequences because it's ligated to the longer strand of DNA. The short ones are down in the 30 mer range, middle length in the 40 mer range, and we get all the way up to the 80 to 90 mer range. These all show up in the base sequences of individual molecules passing through the nanopore. Christian Meyer, I think that this answers your question. Wet-dry cycles can drive polymerization of non-activated mononucleotides. If you put some base pairing nucleotides, D, AMP, and TMP, through multiple wet-dry cycles in the presence of a very simple lipid mixture that can form membranes. That's a mixture such as Sarah talked about in her first talk in this session. You can see the membranes in the panel under A. Those are self-assembled membranes of that lipid mixture. After one cycle in the presence of the paired DAMP and TMP, you can begin to see fluorescent material appearing inside the vesicles. 2 cycles, 3 cycles, and after 4 cycles, you can see what we presume is the DNA stained by the DAPI stain that is accumulated within these lipid membranes. Last question, can we visualize the polymers? With Zoe Hassenkam, we're running atomic force microscopy. We expected to see particulate structures of these small sequences of RNA or DNA. We're working with RNA here. We expected to see little globular particles, but we were surprised to see some ring-like structures. And this is expected by the experts around Christian Murdy. When I talked to him, said, well, yeah, RNA strands, short ones, do have the potential to cyclize. And I think that we're probably seeing some ring-like structures appearing. Here's a whole collection of these that Tue collected for a publication that appeared last year. Do this both with GC and with AU, but not with single nucleotides as monomers. Here's my last couple of slides. I really like the idea of taking our laboratory experiments out into the wild and seeing whether this works in these prebiotic analog sites. So what we did was to go to a hot spring in the North Island of New Zealand called Hell's Gate. And you can see them boiling away here. This is a very active hot spring near Rotorua. We had our mixtures of mononucleotides and lipids in those little vials you see down there in B. And these were then put down into the water of a hot spring, just a little over 90 degrees C. And we used the water of the hot spring as the water to go through these wet-dry cycles. I then took that back to the lab early in last year. That was interrupted by the COVID when it came through California in February. But we did get just some preliminary results. So I can show you that in the laboratory simulation, we can see our product by ethidium bromide staining, and we see that same product in the polymers isolated from the hot spring conditions. We can test how much we made, the yields of these precipitatable ethanol precipitation of polymers using a nanodrop spectrophotometry. And we did this experiment the first time in 2018 and repeated it with more specimens in 2020. And there's something interesting. You can see that the uridine mononucleotides down in the lower portion of both those figures had higher yields, and that was repeatable. The adenine had lower yields, and we did get these polymers precipitating in both experiments. So I presume that our experiment really does work, not just in the laboratory, but also using these prebiotic conditions. So to sum up, this is an alternative scenario for life's origin. We suppose that life began in freshwater hydrothermal pools subjected to wet-dry cycles. The conclusion is that life did not necessarily invent nucleic acids. Instead, the chemical energy made available by wet-dry cycling generates random nucleic acids, such as the RNA-like polymers that I showed you here. These get encapsulated if amphiphiles are also present, and we end up with what we call protocells. These are vast numbers. Every time we do the experiment, we make trillions of these protocells. Each one is different from all the rest. So in a sense, each one is a microscopic experiment. Because they're all different, they can undergo selection and evolution. This is the edge of where we are in our work. We are now trying to think of ways to get these to undergo selection and evolution. So this is a proposed alternative to be tested, that life began when selected systems of encapsulated polymers emerged that were capable of metabolism, growth, and replication. Okay, that finishes up my talk. And I will get out of screen sharing.